Hello students. So, today we are in the last lecture. We have discussed many technologies, different ways of looking at various type of interaction analysis, both label based and label free platforms. We also discussed about how to really start analyzing the data and visualizing the data. And finally, we have to think about how best all this omics information which we are obtaining, how this could lead to the understanding of the physiology. As you can see the slide, that we are generating data set from variety of different level of biomolecules, starting from the genome, transcriptome, proteome, and then looking at this whole information in the more systems network manner, which eventually can help us to define the physiology and probably help the life scientist and medical scientist to really provide the meaningful information for the complex biological questions. The complex data sets which we are obtaining from different technologies, different platforms, different omics levels, we have to provide a much more comprehensive view of what is the meaning of this data set. And that is where systems biology and integrated omics analysis have really started leading the path of this whole field, and these systems approaches are really required to analyze and interpret these large data sets, which will eventually help us to find the mechanisms of different diseases, mechanism of different complex biological problems and various therapies for diseases. It may also help us to relate the different phenotypes and what could be the relevance to the clinical characteristics if we are looking at more medical issues. So, what is system biology? System biology is systems level understanding of the biological networks, which is defined with the network of various interacting elements as well as the dynamic perturbations and responses to the various cues which we obtain from those systems. These networks provide the insights which cannot be analyzed just from the isolated systems alone. We cannot just look at the genome separately, transcriptome separately, proteome separately and try to obtain the full picture. We have to start putting the things together in a more systems wide manner, which could be analyzed using systems level tools. So, what are the common elements of system biology? They include the networks, modeling, computation and dynamic properties. So, what are the approaches of system biology? The system is an entity which maintains its existence through mutual interaction of its constituent parts. You can see on the slide that system research consists of first identification of the parts, then characterization of these components, excluding the ones which are not the part of a system, and then identifying the interaction of the components with each other. Finally, we are looking at the interaction of these components with environment, which modulates the parts either directly or indirectly through modulation of these internal interactions. So, what are distinct approaches in system biology? It can be model based or data based. In model based, you already have the prior data information which you want to implement and build the model. Of course, it relies on a lot of computational modeling and various type of tools for simulation, but it is very difficult to build the detailed kinetic models just based on this information. Whereas, the data based system biology approach involves a new phenomenon to define or find using these kind of data sets. It relies essentially on various type of omic data sets and looks at their intricate complex relationship by looking at various components from genome, transcriptome, proteome and then try to define various type of pathways and networks. There could be different ways of looking at the system biology approaches. One is reductionist approach, which is understanding the properties of complex systems by studying their fundamental parts in its individuality, or it can be integrative approach where we want to understand the properties of the fundamental parts and their interactions to predict the system. What is system biology triangle? It involves the experiment which we do in the wet lab to generate the data set, the various technology platforms and computational modeling. All this together constitutes the system biology triangle. This is essentially the synergistic application of experiment, theory, technology and modeling to enhance our understanding of the biological processes as a whole system rather than looking them as the individual components. So, I must warned you that systems biology field is very challenging, because you are looking at not only one type of data set in isolation, but you are trying to integrate the information and looking at the properties of a system in its whole, which is not just simply adding 1, 2, 3 and making the sum of that, but looking at much more bigger picture and trying to integrate the information in much more meaningful manner. And when you do systems analysis, you might find out that system may have its own emerging property on its own, which was not just possible by looking at each pieces separately and just by combining them together. Understanding the dynamics of even simplest biological networks, it requires lot of computational power using modeling, simulation and understanding of the biological questions. It also requires mathematical modeling and different type of statistical approaches. I have depicted you here one of the omics data set obtained from the microarrays and mass spectrometer, and then how we were trying to build the interaction networks of various proteins which were involved in a given cancer. So, by looking at this kind of information, we want to really obtain what are the important nodes which might be governing the disease, which was otherwise not possible just by looking at the list of the protein and what kind of proteins are changing, but rather now we are comprehensively analyzing the entire data set and looking at which could be the major root cause of the disease and can we identify the right nodes. If we identify those, the major area, then of course one could think about certain therapies which could target those nodes, and then probably those will be the root cause where one could start making more impact for the patient treatment. So, in my next part I am going to now talk to you about some of the major revolutions which are happening in front of us in the field of omics. I am going to give you some very brief examples and also going to give the perspective directly from the experts, the scientists who are working on this field and who have actually laid the foundation for these areas. So, first in the field of interactomics, the big project has come forward, which is the Human Protein Atlas. The scientist Matthias Ullhen and his big team, they are all working towards generating antibodies for each protein and looking at that how the proteins are localized, how proteins interact, and they are providing lot of interesting biological insight looking at the proteins by different type of experiments which are based on the antibodies. So, let me have a perspective of Dr. Matthias Ullhen and Dr. Emma to talk to you about what are the goals of these big project of Human Protein Atlas and what are the major accomplishments so far.
So we have during the last 10 years as part of the Human Protein Atlas project, we've been making a cell atlas, a subcellular map of the human proteome using an antibody based approach. So we generate high resolution fluorescent images, and we've been generating hundreds of thousands of such images, and then we've been classifying the patterns and sorting it into different categories so we could finally provide the first subcellular map of the human proteome and see which proteins are in the mitochondria, which proteins are on the plasma membrane, and so on. And the most interesting findings we made here is that as much as half of all proteins localize to several places in the cell, and we also see a lot of single cell variations. So the multilocalizing proteins, as we call them, it's very interesting from a biological perspective. We don't know the biological consequences of this. It might be so that these proteins have context-specific functions or even moonlight in different parts of the cell, but we can't really tell. We can only make the observation that they are in multiple places, and then someone else would have to do in-depth biological studies, look at splicing isoforms or post-translational modifications to see if it's how this multilocalization is achieved and also what the biological consequence is. My opinion is that antibodies should be viewed upon as small chemical reagents. They have on and off target binding, so you have to validate your results in any assay where you use them. So we fail more than half of the antibodies that we use, so we put a lot of effort into validation of our data. And we have validation scores that kind of denotes the reliability of the results. So that's a big part of our job.
Next, I'd like to talk to you about the field of proteogenomics. While a lot of things we talked about different proteomic technologies, but you have also seen that how NGS is contributing, next generation sequencing, and genomic field is contributing immensely towards our understanding of the biology. So, how to integrate this information? Can we start integrating the most crucial molecules like genome and proteome together, and that has actually led to the field of proteogenomics, which is essentially going to look at a lot of information which was not possible otherwise by looking at the molecules alone. Probably with this cartoon you will appreciate that how the proteogenomic analysis of cancer and other complex diseases could elucidate the functional consequence of somatic mutations, narrow the candidate nomination for the driver genes which may have the large deletions or amplified regions, and probably identify the therapeutic targets. The National Cancer Institute and National Institute of Health in USA, they have really made huge contribution in this area of proteogenomic investigations. There are a series of interesting papers which I have shown on the screen. They have shown the utility of integrating these technologies and how one could actually understand the very complex cancers in much more efficient manner, especially the studies on the colorectal cancer, breast cancer and ovarian cancer have really given the path that how to integrate this information and get much more meaningful insight for the same complex disease which was otherwise not possible. So, in the field of proteogenomics, which actually we offer another course on this is a much complex subject and requires much more discussion, but to give you a flavor, it involves from the same sample and it is more used in the case of cancer and other complex diseases. From the same patient sample, can we look at DNA and look at their whole genome sequence and whole exome sequence, then look at the RNA analysis, RNA sequencing, entire transcriptome analysis and protein and analyze using LC-MS, and then start aligning the data, start analyzing the data, big data to obtain the proteogenomic information which has given those papers which I just showed you in the previous slide. There have been many beautiful studies which have shown the impact of these studies, and they have also shown that how if you build the layer wise information, as you can see in one of this nature study shown in the breast cancer, that if you are looking at different subtypes like basal like, HER2, luminal A, luminal B, these cancer type, if you are only looking at one layer of information only at the gene level or even only the protein level, that may not help you to obtain the entire picture, but even sometime information at the phosphorylation level or PTM level is much more powerful than just looking at all of this other molecules. So, you have to get an idea that how this entire information can be put together and can give us much more meaningful insight. So, based on this idea that how to utilize these omics field, omics technologies, the ex-US Vice President Joe Biden, he started the project Cancer Moonshot. Idea was, can we accelerate our pace of doing cancer research, and whatever we are able to achieve in next 20 years can we achieve in 5 years. So, in this manner scientists really got motivated and started coming together in the field of proteogenomics, which led to the Cancer Moonshot USA program. The next intention was, cancer has no boundaries. Can we expand this project to the international community, and they formed International Cancer Proteogenome Consortium to have more association and collaboration from other countries. We are very fortunate and proud to be part of this program of ICPC, where India joined the 12 country consortium, and we are also going to look at the proteogenome analysis of different complex cancer of India, and then share the data and analyze the data with different countries and try to obtain more meaningful information for these difficult to treat cancers. An image shown here for one of the ICPC retreat where different countries are participating in this activity. So, here in the picture I have shown Dr. Henry Rodriguez. He is one of the director of NIH, NCI, who has really led the path for the ICPC program, and I am going to show you a video clip of sharing his perspective about the ICPC program and Cancer Moonshot, as well as what are the guidelines for doing research in this area and the way forward, his vision.
So what I could speak to is the one from the National Cancer Institute, which is the effort that I lead. I think the part that's very exciting is have watched now the evolution, as I would call it, and the maturity of the science of proteomics. So 10 years ago, when we started this effort at the Cancer Institute, referred to as now the Clinical Proteomic Tumor Analysis Consortium, CPTAC, there was an acknowledgement out of NCI. While we saw the potential for genomics, we knew proteomics would be a very complementary field. But at that time, we felt the technology that was being developed, in this case mass spectrometry, yet while it is extremely a very powerful technique, there were items that we needed to address in terms of the rigor and reproducibility if it were to one day be a complementary technology to the genomics landscape. So 10 years ago, while the Cancer Genome Atlas was the very first one, large-scale biology, that we moved forward in cataloging all the perturbations inside cancerous cells from a genomic perspective, the proteomics field, we decided to take a little different approach. We went after the analytical rigor of those technologies. It took about 5 years. But once we got that finally on hand, the question became, what would we do next? There was the opportunity that we seized. We went after those same tumor types that the genomics community went after in TCGA, and we applied those proteomic-based technologies, mass spectrometry, onto them. When we saw the beauty of the additional biology that was starting to be generated from the combination of these two disciplines, that's when we saw a lot more potential. So today, this program now, in its next iteration, the ways it will be expanding, we will go after additional cancer types from the first program. So the first program we went after colorectal, breast, and ovarian cancer. Now we will be cataloging approximately additional 5 cancer types. But the most exciting part that I would have to say, for the first time, we're going to be teaming up a proteomics laboratory expertise with an ongoing NCI-sponsored clinical trial. And these clinical trials are typically genomically driven, but we recognize that genomics is still advanced, but there's an opportunity to fill in the void of the biology of trying to understand why patients either respond well or do not respond well based on a genomic-driven information to the treatment that they were just administered to. I'd say the best way that I would look at it is, what is driving precision medicine today? In oncology, which is my specialty, I think clearly genomics is making tremendous strides. There are exquisite examples today where we're able to identify and be in a very great position on how to treat a patient simply relying on genomics. However, you gotta flip your coin. There are still many instances in oncology where we do not fully understand the biology and how these individuals are responding to the treatments. That's where I see now proteomics filling a very critical piece of that information. So what we're trying to do at the NCI through our initiatives is to begin to converge these disciplines. And quite frankly, partly I think the reason has been is the evolution of technology. Genomics technology has matured and is still maturing at a faster rate. But as proteomics technologies are also maturing, and also we could go after smaller samples, it's just a matter of time that these disciplines do begin to converge. So that's why I see these two areas being very complementary to one another, specifically in the area of precision-based medicine for oncology. As opposed to the common problem of mass spec, I would say what's needed, because I really don't see them as problems. I just see them as the evolution of technology. My one wish, I think, which is a natural wish that you have of all technologies, a common one, you want automation. You want it to be very simplistic for the average user to use. You also want that the system, as technologies evolve, to potentially using less sample material up front. But keep in mind, I think this question is one that people tend to ask on is, well, how much material do you need to do the analysis that you wish to go after? The question that people should be asking is, what is the question you're trying to ask of the biology? That question will dictate what you want the technology to do. And in most instances, you find out when people really begin to ask the question they would like to see of the experiment, technologies themselves are quite mature. From what I've seen from my perspective, proteomics, the way we know it today, would not exist without mass spectrometry. Pure and simple. Genomics gave us a blueprint. What people did within the specialty is, there was an acknowledgement, here is a mass spectrometer. Mass spectrometry has been used in clinics for over 30 years for different purposes. It's mainly used for small molecule screening. But then there was a brilliant idea that came into play. Can you take a big protein, break it apart into small pieces, into these peptides, and informatically, just like an eloquent puzzle, stitch it back up into a protein? Without mass spectrometry, proteomics as we know it today simply would not exist. So the cancer moonshot, I'd say, is one of the most incredible things that's been developed off the United States over the past approximately 1 year. So obviously, one of the main drivers here is the 47th Vice President of the United States, Joe Biden. It's been a huge honor to be part of that effort. Two programs that I could specifically speak to myself on. One of them we actually did, which is a partnership between the National Institutes of Health, specifically NCI. Then we partnered with the Department of Defense. We also partnered with the Veterans Administration. This program is referred to now as Apollo. This effort, along with another program that we've now launched under that Cancer Moonshot umbrella, is an international program. This one now is referred to as the International Cancer Proteal Genome Consortium, or ICPC. That program now, which is an incredible effort in itself, actually now involves 27 institutes, spans 11 countries. What's nice about it, it fills one of the key promises that our former vice president wanted, and that is the data that we generate in this international effort will be placed in the public domain. But the part that's nice about these two initiatives is that each one of them, ICPC and also the Apollo, that will also be giving up the data in the public. But what brings them together, it is the foundation of what CPTAC actually did, and that is a recognition, converged genomics with proteomics, with the hope that not only you will identify new biology, but as in the Apollo case, they really want to push it much more faster in the translational space, and potentially in the near-term future, begin to apply it towards patient care.
Another interesting development happening in the field is integrative personalized omics profiling. Essentially, this path was led by Dr. Mike Schneider from Stanford University, who took his own samples and analyzed longitudinal manner using various multi-omic technologies, which really led first time idea that he might be prone for diabetes, which was detected clinically very late, but looking at the biomolecule they could sense this much ahead of time. And this really constitutes that how looking at the multi-omics analysis from the same individual can provide more meaningful information. And likewise, there are many success stories, many different case studies are now coming forward, which are showing the utility of looking at personalized omics profiling. There are also groups in Michigan who are looking at Michigan Center for Translational Pathology, Dr. Arul Chinnayan and team. They are doing from the same patient looking at their genome data, their proteome data, their metabolome data, how one could start finding the right cure for the given tumor type, which is otherwise not possible, and they are able to provide at a much comprehensive level this kind of analysis and directly reaching to the patient care. Now, Dr. Mike Schneider and his team has launched a major program on human personal omics profiling, or HPOP, and idea is to provide much deeper understanding of the genetics of the human body and provides the new clues of different diseases which was otherwise not possible. Let us hear Dr. Mike Schneider about his program and vision for the HPOP project.
What is the role of the HPOP project? The HPOP project is really trying to understand what it means to be healthy and how do people differ all around the planet. So what we're doing is we're actually taking people's blood and urine and also their stool samples, and what we're doing is doing deep omics profiling, the proteome, the metabolome, the transcriptome, to make as many measurements as possible to see at a molecular detail how people differ from one another. So we're going to see this, we're going to sample people from all around the globe at these human proteome meetings, and then we're going to see what their molecular diversity looks like, how do different ethnic groups and different locations have an impact on people's molecular composition. Do I think omics will be used in clinical situations? I think at some level, absolutely. That is to say, we'll be making thousands, if not more, measurements in the future. Right now, when you go to a doctor, they make about 15 measurements on you. And in the future, I think we'll be making hundreds, if not thousands, using various omics methods, especially proteomics and metabolomics will be very, very powerful for looking at people's molecular state, which I think will say a lot about their health and also what happens when they get sick.
Let me now take you to the next project, which is wellness project. We have been talking mainly about how to compare the healthy individuals with disease, but can we look at the wellness or the health status of healthy individual or seemingly healthy individual in the longitudinal manner? So, can we look at their entire omics profile at different time interval from 0 time to 6 months to 1 year, 2 years, and start looking at are there changes happening in dynamic manner inside the body which might give us the clues for what disease the individual might be having or whether the individual is still healthy. So, the individual dynamic data clouds has to be integrated, and that field is known as P4 medicine, which is led by Dr. Leroy Hood from Institute for Systems Biology in US in Seattle. This big project of wellness was also published 2 years ago in Nature Biotechnology, has shown the utility, and many similar projects have now started in different countries which are looking at the omics data set to build the wellness. Let us hear the views of Dr. Leroy Hood on the wellness project and P4 medicine.
I am very excited by the possibility India would consider a scientific wellness program starting with a pilot project in its own population. Scientific wellness, I think, is the keystone for the revolution in medicine, because through a longitudinal analysis of populations using what I term personal dense dynamic data clouds, that is, the analysis of genomics, of proteomics, of metabolites, of clinical chemistries, of digital assessments of activity and sleep and things like this. All of these together, when analyzed properly, can lead to actionable possibilities to improve wellness and or to avoid disease. But I think the really transformational possibility scientific wellness presents is that if you create a population of people that follow the scientific wellness program in a longitudinal manner, and if that population begins to reach a certain critical level of thousands, you then begin to see wellness to disease transitions that you can use the dense dynamic data cloud to get biomarkers to mark, say, the earliest transition for diabetes. And then you can begin to think about the systems, technologies, and systems-driven strategies that can be used to identify the disease-perturbed networks that will reveal candidates for creating drugs to reverse these diseases at their very earliest stage. And if, for example, over a 20-year period, we can reverse all major diseases before they ever really manifest themselves as a disease and become irreversible, will save the healthcare system an enormous amount of money. In the US, for example, 86% of our enormous healthcare budget goes to chronic diseases. Suppose in 20 years, there are very few chronic diseases left. We've transformed a healthcare system that thinks only about disease to a healthcare system entirely focused on optimizing wellness for their population. This is the opportunity that India has, and I'd be delighted to help you in that endeavor if you decide to make the commitment.
So finally, coming back to the proteomics, and the field of proteomics is now very well led with the efforts of Human Proteome Organization, which tagline is translating the code of life. Idea is many scientists working in the field of proteomics and integrated omics, how to come together, will start sharing the data, will start sharing the ideas, exchange their various information, build collaboration and come forward for the big projects. And Human Proteome Project is one of the ambitious projects which is still happening, which is looking at the chromosome centric Human Proteome Project or disease and biology centric Human Proteome Project, and now there are new pillars added for the pathology and data driven Human Proteome projects. So, let us hear the detail of HPP project with ex-president of HUPO, Dr. Mark Baker.
The Human Proteome Project has two major goals. The first is to map the expression of the human genome in terms of which proteins are made and which post-translational modifications are on those proteins that affect its activity. And the second is to springboard discovery in new drugs discovery around biomarkers for human disease, and an understanding of human health and wellness. One of the major activities of the Human Proteome Project has been to determine how many of those proteins we've observed in human health and human disease. So far, we've taken an approach that looks at about 85% of the total proteome content of the human proteome. But out of the 15% that's missing, about 7% of them, we have some evidence for them. That's either mass spectrometry evidence or evidence that comes from other types of scientific experiments, biochemistry, physiology, pharmacology, genetics. But there are still about 1,000 proteins in the human proteome that we have absolutely no scientific evidence for their existence whatsoever. Proteomics is not a word that everyone in the street knows. If I was to explain what proteomics was, it's effectively the protein complement, or the machines that are made from the human genome, from the coding instructions, into something real that makes a human body work. So the proteins are those things that are important in the machinery of life, in the reproduction of life, and in the development of disease. That's why it's important for us to understand the proteins as well as the genes, in order for us to understand human biology and how that goes awry in human disease.
Additionally, it will be interesting to also listen the last 3 HUPO presidents, Mark Baker, Mike Schneider and Stephen Pennington, and hear from their perspective what proteomics and HUPO has achieved so far and what are their vision for the future guidelines for doing research in this area. I hope this will be an interesting conversation for you to know the perspective of these leaders who are leading the field of proteomics. Okay, so finally I will stop here to show you this slide, which is a motivation for many people working in this field of omics and life sciences, where a lot of the actual problem, we want to find the cues and the mechanism and the leads, understanding them at the omics level, at the molecular level, and then want to translate our bench side findings to the bedside for the actual patient care or for the actual biological system to really make impact of that. And that is where all of this understanding, all of this technologies are really going to help us to achieve that major goal. I hope this whole course, the different type of live sessions, different demonstrations were helpful for you to appreciate the power of these omic technologies. It has definitely given you more confidence that you can start doing many of these experiments and projects even in the small colleges and other universities where you may not have availability of these big gadget and platforms. With the access of so much data available being shared by the entire community, I think you can start looking at and appreciate the data in a very different manner. I hope the whole course has given you more understanding, much more new knowledge about the field of interactomics, more in detail, as well as the broad understanding of the field of proteomics and other omics sciences. Also, you are now more confident to start doing some experiment and doing project in this field. We will be happy to have more interaction with you, which we will be doing further, and I hope that some of these understanding and knowledge is going to help you not only in your research, but also in your future projects and for your career. Thank you very much for attending this lecture and attending this course, and wish you all the best for your future endeavors. Thank you.